Welcome, everyone. It is 1 o'clock, and we will go ahead and get started. I'm sure some people will continue to join us. My name is Laura, and on behalf of the Dementia Friendly Coalition for Brown County, which is part of the Dementia Friendly Communities Initiative, we would like to welcome you to our latest research presentation today on environmental impacts on brain health. Before I get started and introduce our presenter, I'd like to do a little housekeeping. I just want to make sure everyone's aware that we are recording this presentation today. We will, for everyone who registered, the Alzheimer's Association has graciously offered us this platform to use and to do the recording. They will be sending out a copy of the recording so you can go back and listen to this great presentation. So we'd like to thank the Alzheimer's Association for all of their help. Also the Forget Me Not Fund, which has helped us in some of the funding for this program. So instead of listening to me, I'm going to introduce Dr. Renee Richer, who is with the University of Wisconsin-Green Bay in the Marinette campus since August 2014. Dr. Richer has spent a last decade researching the relationship between environmental quality and human health. In particularly, she studies the relationship between water quality and the neurodegenerative diseases such as ALS, Alzheimer's, and Parkinson's disease. And I believe she's found some correlations from some of the other areas that she studied in some of all of that water we have around us here. So, Dr. Richer, I will let you take over.
Great. Thank you very much. I'm so pleased to be here and to join you. As Laura said, I'm at the UW Green Bay Marinette campus, but I'm a fifth-generation Yooper from Gladstone, born and raised in Gladstone, and have managed to do work in all sorts of interesting environments around the world, and then returned home to the area to raise my family and invest here in the legacy. So I'm so glad to be back in the area where I was raised. I just want to point out before we get started that northern Wisconsin is an area which has a disproportionate number of cases of neurodegeneration, in particular ALS. So some of the first ALS hotspots identified in the United States were in this northeastern Wisconsin area. So we'll look at some of the reasons why that might be and what factors might be contributing. And I very much look forward to your questions at the end. So I'd like to give you an introduction into the research work that I've been doing in the last 15 years or so, and how that applies to issues of brain health here in Wisconsin. So on the first slide, you see from Appleton to Arabia. And so we'll be going from one of the hottest, driest places in the world to obviously one of the areas which has the greatest number of lakes. And what is the tie between these two areas? The tie is neurotoxins in the environment. So we'll talk a little bit about that as we continue on. So I want to start out first with a very important story. And this story about the Chamorro people of Guam is what got scientists first thinking about environmental causes of neurodegeneration, in this case specifically ALS. So up in the left-hand corner, you can see the red arrow is the island of Guam. And just after World War II, researchers, doctors were finding that 25% of the population, up to 25% of the population, was dying as a result of a neurodegenerative disease that looked very much like ALS with a mix of Parkinson's in it. And so they did a tremendous amount of work looking at what were the potential causes. And it turns out that the Chamorro Chamorro people were eating a diet. They made their flour from this interesting plant called the cycad, which you see on the far right-hand side there. So they would collect the cycad seeds, they would pound the cycad seeds, and they would use that as flour. It was one of the staples in their food. Also, you can see in the picture of the cycad, you see what's called a flying fox, but it's actually a bat, of course. And that bat is a great delicacy. And so the bats would also consume the cycad seeds. But it turns out that cycads had this neurotoxic amino acid in it. So let's go down to the lower part of the image there. And these are sections of root from the cycad plant, and you can see they're green. And that green component is actually cyanobacteria, which are photosynthetic bacteria. And those bacteria produce this neurotoxin, this neurotoxic amino acid. We'll talk about it in a little bit more detail. But that's what's illustrated there on the far right-hand side, L-BMAA. So in the late 1940s, early 1950s, they saw that there was this link between neurodegenerative diseases, this tremendous death rate from neurodegeneration, and the diet that the people were consuming. But they didn't have the mechanisms. They didn't understand why exactly it was causing a neurodegenerative disease. So let me just give you a little introduction into what are cyanobacteria. So cyanobacteria, cyan means blue or green, and these are photosynthetic bacteria. So these are the organisms that we saw on Earth roughly 3.5 billion years ago, produced oxygen in the environment. We used to call them blue-green algae, but they are not algae. They're actually photosynthetic bacteria. So on the left-hand side of the image, you can see that we have blooms of cyanobacteria in our freshwater systems. And this is becoming an increasing problem in the Great Lakes area, and in particular to climate change. So we'll talk about that as well. What is the role of climate change in fueling these cyanobacterial blooms? On the left-hand side, you'll see an image from Qatar, which is a small country off the Saudi Arabian Peninsula, where I worked for 8 years looking at cyanobacteria and the toxins they produced. And what looks like mud in the foreground there is actually this mat. It feels a bit like leather of cyanobacteria. It's a very complex bacterial community, but it is actually cyanobacteria. So cyanobacteria love extreme environments, and they also love fresh water, they love salt water, but they produce some of the most potent toxins known to man. So I'll take you to Qatar and talk briefly about the research work that I did in the Middle East, and then tie that back specifically to what we see in northern Wisconsin. So you can see that the state of Qatar is a little peninsula off of Saudi Arabia. It is incredibly dry. They get anywhere from 50 to 100 millimeters of rain a year, and you can see what the landscape looks like. So those are 3 of my students, and we would go out into the field and do research. So it's a very sort of a dry, barren type of landscape. But because of the extreme conditions, one of the few photosynthetic organisms that thrive are cyanobacteria. So in fact, the major photosynthesis going on in this type of environment is not by plants, but by the cyanobacteria. So it's so important in these desert areas. So I asked a very simple question. In the state of Qatar, I said, well, where are all the cyanobacteria? If it's so important in the ecosystem, where are all the cyanobacteria? So we did a survey of roughly 90% of the entire land area of the country. And we saw that in the northern regions, as much as 90% of the land area was covered by these thick bacterial mats. So that said to us, well, there's a lot of potential then for people to be exposed to these types of toxins, cyanobacterial toxins. And I just want to pause in the upper left-hand corner of the slides, of the images. Those are the original scientific papers. So if you have interest in going back and looking at the original work, that will show you the title and where that research was published. So if you're not familiar, that's what that is in the upper left-hand corner. That's the original citation for the research. So what we found is that there was a lot of variability where cyanobacteria were in the country, related particularly to rainfall, but it sure dominated. And then we said, well, okay, what toxins are actually present then in the country? If we have a lot of cyanobacteria, and we know that cyanobacteria produce these really potent toxins, what toxins are we finding in the country? And so we identified 5 toxins. Microcystin LR, this is probably one of the best known cyanobacterial toxins. It's been well documented in the waters of Wisconsin. And that is it's a liver toxin. It is a tumor promoter, and it has a long history of causing human deaths when humans are exposed either via food or water. Anatoxin AS. Anatoxin, this is what we call an acute neurotoxin, which means it kills you really quickly. And in fact, its common name is very fast death factor. And its structure is very similar to chemical weapons. So we see it acts very much like a biological chemical weapon. And then the last 3 toxins, these are the ones that we'll focus on when we're talking about waters in Wisconsin. BMAA, this stands for beta-methylaminoalanine. It's a modified amino acid. But the important point is that this is the key component of what the Chamorro people in Guam were consuming. This was the toxin that was in the cycad flower and in the flying fox. And this is the population that had as much as 25% of the population dying from these neurodegenerative diseases. The other 2 toxins are just isomers, meaning they're essentially a modified structure of BMAA. DAB is diaminobutyric acid. AEG is amino ethyl glycine. You don't need to worry about the names, but just know that those last 3 ones are important neurotoxins that we identified in the environment being produced by cyanobacteria. And when we look at Wisconsin waters, we will see these exact same neurotoxins being produced in our Wisconsin waters. All right, so once we started and we had identified these toxins, and again, I'm working with a team of people. This is not obviously all my work, and you'll see that in the citations up in the left-hand corner. We said, okay, we know where the cyanobacteria is. We have identified what toxins they produce. We want to know now where are most of those toxins found. And so we looked at it in the soil, and we dug a lot of soil pits. And what we found was that these toxins were accumulating in the soil. So that's what that little image on the left there illustrates, that we found BMAA, AEG, and DAB, the 3 key neurotoxins, which would accumulate in the soil. Then we said, well, if it's in the soil, is it also in the water? So we did a comprehensive survey. We looked at water supplies in the urban environment as well as in the rural environment in Qatar. And what we found again was that we have AEG and microcystin in the water supply. So again, AEG is this neurotoxin. Microcystin is the liver toxin. Both of these we will also find in Wisconsin waters. And then we had fate step in. So in the image illustrated here, you can see my colleague, Dr. Aspasia Hadsiektimu, and my young daughter, and our companion dog, Star. And we were out in the field collecting samples, and Star, the dog, decided that she was going to have a drink of water. Well, it turns out that rainwater she drank from was sitting above a cyanobacterial mat. And shortly after she consumed the water, she started to show signs of acute neurotoxin poisoning. And because this was in fact our area of research and what we were doing, we recognized it immediately. And this picture was actually taken on the day and at the site. We got Star into the vehicle. We drove as fast as we could, and we were able to get her to the vet, and she survived that acute poisoning. Well, it turns out that there were other animals that were dying, but it hadn't ever been attributed to neurotoxins in the environment. So vets were having dogs come in with these symptoms, and they thought they were dying of overheating or being exposed to rat poison or other types of toxins. So because of this unfortunate incident, we were the first to identify cyanotoxins in desert environments as a cause of poisoning in domesticated animals. So it was just that unusual stroke of, I wouldn't want to say luck, but we just happened to be in the right place in the right time, and everything turned out well. But we now know that after a rain event, if you have cyanotoxins in something as innocuous as a rain pool, you can kill a mid-sized dog, let alone small children that might be playing in the rain pools as well. So that was something that was unexpected that came out of our research. So we'd identified the neurotoxins in the soil. We'd identified neurotoxins in the water. We had identified neurotoxins after something as common as a rainfall event or uncommon in the desert. And then we said, well, what about the food? If we're consuming a lot of fish, and this is an area where people would consume a lot of fish, are we finding those neurotoxins in the food? And so again, we did this comprehensive survey looking at the key fish species that were consumed in the area. And we found, yes, in fact, that these neurotoxins were accumulating in the bodies of the fish. So diaminobutyric acid, amino ethyl glycine, we found those accumulating in these marine fish, which are key source of food. So what are some other ways people might be exposed to neurotoxins? And in the Middle East, sandstorms are as bad as this picture illustrates. It is as if there is a wall of sand that suddenly encompasses you. And you can be in a sandstorm for up to 5 days. We've had some really unpleasant sandstorms. But what we found was that these neurotoxins, cyanotoxins, were also in the air. So we ran a few numbers, just looking at the concentrations that we found in the air. and how often people breathe. And in a sandstorm like this, within 24 hours, you could exceed, the tolerable daily limit for exposure to cyanobacterial toxins. In children, that would be even worse. We know that children, because their body size is smaller, because they have a higher metabolic rate, that exposure to toxins poses a larger load, a greater risk for them. So I know you're thinking, Renee, here you're talking about a sandstorm, but we are in Green Bay. The point is that these toxins can be aerosolized. And we are finding great evidence now coming out from Australia, from the United States, from Europe, that aerosolization of toxins from water bodies is being linked to hotspots of neurodegenerative illness, in particular ALS, Lou Gehrig's disease, amyotrophic lateral sclerosis. So we also have to think about them, cyanotoxins, moving through the air. And the last point I want to make about the Middle Eastern Qatar is that this neurotoxin, BMAA, which we identified in the environment, thus far remains the only reasonable biological exposure agent to account for Gulf War syndrome. So in the first Gulf War, we had soldiers going to the Middle East. They were exposed to the desert crust, which is made up of cyanobacteria in these desert areas. A lot of that would have been aerosolized, as you can see in the images. And so we made that potential link between Gulf War syndrome and exposure to cyanobacteria neurotoxins. All right, so the work that we did in the Middle East then, what did we find? We found that there were neurotoxins, cyanobacterial toxins, in the soil, in the water that people were drinking, in the food that we were eating, and in the air that we were breathing. And we will find these same cyanobacterial toxins in Wisconsin, in the water that we drink and swim in the food that we eat, and in the air that we breathe. And again, there's a fair amount of research looking at exposure from lakes that have cyanobacterial blooms. So let's look a little bit more now at how do these neurotoxins cause disease, and look at Wisconsin as well. All right, so what is the link with Green Bay? I've been talking about this very desert area in the Middle East. Well, the link with Wisconsin and Green Bay in particular is harmful algal blooms. So we know that the frequency of harmful algal blooms is increasing. We know that the duration of harmful algal blooms are increasing, and the intensity of those algal blooms are increasing. So algal blooms, and I'll show you an image if you're not familiar. This is when you have this great bloom of photosynthetic material. They are called algal blooms, but again, the main component is cyanobacteria, and that comes from that historical influence with calling cyanobacteria blue-green algae. So when we talk about harmful algal blooms or toxic algal blooms, the key component there is our photosynthetic bacteria, cyanobacteria. So you may have seen this, right? So Lake Erie, Saginaw Bay, the water can suddenly overnight look like pea soup. And what this is it's a sudden growth of cyanobacteria in the water. Lake Erie is disproportionately impacted, partly because you have a lot of industrial waste, nitrogen, phosphorus, but also because there are areas of the lake that are more shallow and more warm. So the areas surrounding that western part of Lake Erie often have cyanobacterial blooms. Right now, there are 3 freshwater bodies in the United States that NASA is monitoring from space for the outbreak of a cyanobacterial bloom. And Green Bay is one of those sites. There's another site in Florida. There's another site in California. So Green Bay is being monitored again from space by NASA for the outbreak of these blooms. They're looking for ways that they can predict a potential bloom to prevent it and protect people's health. So again, harmful algal blooms are in fact a sudden growth of what is largely cyanobacteria, and then they can produce toxins. Okay? Under certain conditions, they will produce toxins, but not under all conditions. All right, so what toxins do we find in Wisconsin waters? And Lake Winnebago in particular, Green Bay is also a good example. We find our liver toxin, microcystine, that is the most well-known, probably the best studied. Anatoxin, this is the acute neurotoxin that almost killed the dog and did kill multiple dogs. And in essence, it is like being exposed to a chemical weapon. The structure is very similar. And our 3 neurotoxins, BMAA, DAB, and AEG. So those have all been identified in Great Lakes waters and in the Wisconsin area. So I want to spend a little bit of time looking at the one neurotoxin, BMAA. So BMAA, you can see the structure on the lower right-hand side there. It is a modified amino acid. Now, all of our proteins, the structural part of our bodies that perform all of the functions in our bodies, that is comprised of proteins. And the building blocks of our proteins are amino acids. So the proteins in our bodies are comprised of 20 basic amino acids, 20 amino acids. It turns out that the neurotoxic amino acid BMAA tricks the body into thinking it's serine. So let's just look at this image here. So we have 20 amino acids. They form chains of amino acids to form our proteins in the body. And we have 20 amino acids which are used to make the body's proteins. And our fast train on the bottom there, you see L-serine. This is one of the typical amino acids used to construct our bodies to make our proteins. But it turns out L-BMAA, or BMAA, this is the neurotoxin produced by cyanobacteria, is modified. But the body mistakes it, right? The machinery in the cells mistake it for serine and incorporates it into our proteins. So it's a bit like putting Thomas the Tank Engine in one of our fast track bullet trains here. So you can imagine how that disrupts the protein structure as well as the functioning of the protein. So that's just a short reminder of why amino acids are important and how this modified amino acid can be toxic to the body. All right, so let's go back to our harmful algal bloom. which we can see illustrated here. So there will be a variety of toxins being produced at any one time by a harmful algal bloom. That is most commonly going to be microcystine, the liver toxin, but also our 4 neurotoxins can also be produced. And so exposure to them, whether you are swimming in the water skiing in the water, boating, jet skiing, you could have those toxins aerosolized. So we have found that people who have their houses on the downwind side of a lake, which has regular cyanobacterial blooms, we see hot spots of ALS, Lou Gehrig's disease, associated with living on the downwind side of lakes, which have cyanobacterial blooms. Fish, which are swimming in lakes with cyanobacterial blooms, can incorporate those toxins into the body of the fish. So when we consume the fish, we are also potentially exposed to those same neurotoxins. And at this point, I just want to note that the ALS hotspots that have been identified in northeastern Wisconsin have been linked directly to consumption of fish. That could be cyanobacterial toxins, but it could also be exposure to mercury. Now, it's important to know that mercury is a very potent neurotoxin, and as we see both mercury and cyanobacterial toxins accumulate in fish, we could have a potential double punch with consuming lake fish in regards to exposure to neurotoxins. I also just want to point out, in the last few weeks, the EPA has loosened many of the restrictions on mercury. And so just keep in mind that mercury is a potent neurotoxin. We are now moving into a phase where we are relaxing many of the environmental regulations. And again, in the last couple weeks, the EPA has lessened restrictions on mercury emissions. So that is something that we might expect to see more of in the future. So exposure to the water, which has cyanobacterial toxins in it, consuming fish, which are eating in lakes with cyanobacterial toxins, living or being exposed to aerosolized toxins which are blown up into the air from lakes, are all potential ways to be exposed. All right, so I said that I would outline some of the most recent, and the data is now starting to really accumulate that exposure to BMAA, the key cyanobacterial neurotoxin, is intimately linked to development of neurodegeneration. So the slide that is on the image now, this is one of the groundbreaking work that came out relatively recently. I want to say 2016. And in this case, they developed an animal model. It is the first animal model to show the development of the classic signs of neurodegeneration, neurofibrillary tangles, and plaques as a result of an environmental exposure. So here they have an animal model. The animals were exposed in their food to BMAA, or cyanobacterial toxin, and they developed the classic signs of neurodegeneration, like what we would see in ALS, Alzheimer's, and Parkinson's disease. So that was this tremendous step forward. Up until that point, there had been very little successful research looking at causes of neurodegeneration. And that's because only 5%, in this case ALS, 5% of ALS cases can be attributed to some type of genetic abnormality. So the vast majority cannot be tied to any specific cause, except now. Now we have an environmental toxin that when we expose animals to it, they show neurodegeneration. So this was a fantastic step forward. And my colleagues did this work. I was not directly involved in this work. Now, what has come out in the last 6 months or so? Again, this gets back to the genetic causes of ALS. Few of the cases, 5%, are linked to a genetic mutation in a protein which protects the cell. It's essentially a housekeeping protein that helps to maintain the cell, in particular neurons. And this study, which has just come out recently, showed that when BMAA is incorporated into that housekeeping protein, it causes instability of the protein, and it's less likely to be able to perform its function. So it changes how that protein is able to function. And so again, this was a fantastic bit of work which has come out in the last 6 months or so. This is a paper which has come out again. I think this one came out in the last 2 months. And what this showed is that we saw in animals that have neurodegeneration, okay, that are just starting to show those initial signs of neuronal damage, that they aren't able to produce as much L-serine. And so this should, right, you should be saying, but we saw L-serine before, right? This was the normal amino acid that our neurotoxin BMAA mimics or pretends to be. And so what this suggests is that, in fact, if we consume L-serine, or if we enhance our diet with that amino acid L-serine, that we can greatly reduce the development of those neural pathologies, amyloid plaques, and our neurofibrillary tangles. And if I can just go back a second, I apologize, I want to go back here. In this animal model, where we fed animals, my colleagues fed animals BMAA, they also found that if they fed them L-serine, they were able to rescue them. It greatly decreased the amount of amyloid plaques and neurofibrillary tangles that we saw in those animals. So this amino acid, L-serine, is turning out to be a protective mechanism. And that is where I want to finish. So this is a paper which came out in 2020. So again, I think this came out about 2 months ago. This is authored by my colleagues, which I have worked greatly with. I'm not an author on this paper. But some of the very first signs of neurodegeneration, particularly in regards to ALS, Lou Gehrig's disease, is pathology in the spinal cord. And then it progresses from there. And what they found is that when they fed, again, this normally occurring amino acid, which seems to be intimately tied to neuronal health, when they fed that to animals, they were greatly able to reduce those pathologies, the neurological or neuronal damage in the spinal cord. So again, this is all starting to look as if L-serine, this normally occurring amino acid, when it is limited, we see enhanced neurodegeneration, whether it's due to BMAA or other factors, but that enhancing the amount of this amino acid in whether we do it via diet acts as a protective mechanism. So, these most powerful papers have come out in the last year. In fact, the most powerful 3 have come out in the last 6 months. But the original work came out of Guam in the late 1940s, right? It was that early anthropology work, epidemiology work, which is really what got scientists started pursuing this area of research. So as we move forward, for ALS, Alzheimer's, Parkinson's, which all have some similar characteristics of neurodegeneration. This is a pathway which looks like it's going to be a fruitful area of research. All right. I'm going to skip this. This is another paper which has come out, again, just supporting the idea that BMAA is linked to neurodegeneration in the mechanism by which that happens. So as climate change progresses, we expect to see more harmful algal blooms. So warmer temperatures encourages the cyanobacteria to develop and form a bloom. Increased rainfall is interesting. So as climate change increases, we are seeing increased rainfall in the area. And what happens with the increased rainfall is it washes nitrogen and phosphorus from the land into the lakes. And that nitrogen and phosphorus is really what's fueling the growth of the cyanobacteria. So warmer temperatures encourages growth, increased rainfall encourages growth. Nitrogen, but more so phosphorus, encourages cyanobacterial growth. This is why you're seeing the DNR, the Wisconsin DNR, put a lot of effort into reducing phosphorus into water bodies because phosphorus is the limiting nutrient for growth of cyanobacteria. So the more we can reduce phosphorus going into the water, that's going to help as well. As climate change is increasing, we are seeing more frequent algal blooms. We are seeing them occur over a longer duration and a greater intensity. So we can expect that this is something that we will have to deal with increasingly, and we need to look at better management of harmful algal blooms, particularly given the link with human health. And I just want to end on this slide, and then I'll take your questions. So again, with the increased precipitation in the Great Lakes area as a result of climate change, we're seeing higher lake levels. And those higher lake levels are fueling the release of untreated wastewater into the lake. And the more untreated sewage that gets released into the lake, this is bringing phosphorus and nitrogen into the lake. And again, this is just going to be adding more fuel for these harmful algal blooms to develop. So that's another area that we need to start to think about, which is as we have increased rainfall, how do we prevent the release of sewage water, untreated sewage water into the lakes? And lastly, of course, I have a fantastic number of colleagues and students who are responsible for much of the work in what I presented here. And so I want to make sure that they are acknowledged as well. All right, so can I take any questions? And Laura, should I, how do I escape here from the screen?
If you want to, you can just leave that for right now.
Okay.
We're going to ask people if you have questions, if you can put them into the chat box. We will read them to Renee. If you are on the phone, you can hit *4 to unmute yourself and actually ask in person. And then I'm going to also put in the chat box one more time. We have a survey at the very end. If you could fill that out. Also, if you want a certificate of attendance, you can fill that out to get that. So we do have one question here. The first one says, thank you for sharing your research and giving us your time. How would you recommend we best protect ourselves in these hotspots, besides, I suppose, leaving Northeast Wisconsin?
Yeah, so I want to point out that exposure to cyanobacteria and the toxins they produce is becoming a global problem. So this is not something that we worry about just in Qatar or that we worry about just in Northeast Wisconsin, right? So this is becoming a global problem that will have to be addressed in many different areas. What do we do? If you have a bloom developing, you shouldn't be boating, swimming, jet skiing, water skiing under those conditions. Eating fish, limit your fish consumption if they come from water bodies where you regularly have cyanobacterial blooms. Okay, that's another way, by limiting our exposure via water, via air, or the food that we eat. And I think just being aware and helping to support policies to reduce, for example, phosphorus, policies that will help to limit the effects of climate change, those will help to improve water quality. So just be aware, if you have an active bloom, if you're seeing this water that looks like pea soup, you should not be engaging with that water. Your pets as well. So dogs in particular, they'll go into the water, they come out, they lick their fur, and then they're exposed. So that is the best way to limit your exposure.
Thank you.
So I
Do we
didn't know,
have any
I
other?
thought maybe his diaper was wet, so I started laying him down and.
Oh, Kelsey, do you have a question? A few people have unmuted themselves. Do you have a question you would like to share?
Laura, I went ahead and unmuted. People who have muted themselves are still muted, but if you have not muted yourself, you are now unmuted, so that if you want to ask a question and not type, you may.
Renee, while we're waiting, I have a question.
Okay.
In drinking water that we have, since it comes from some of the lakes that are having blooms, do we have to worry about it in our drinking water that gets filtered and cleaned?
Yeah, so that's a fantastic question. If you remember, I think it was a year or two ago, we had the bloom in Lake Erie, which shut down the water supply for Toledo, in the area surrounding Lake Erie. Now, for most of our water treatment plants, they will look for bacteria, and they will identify microcystin, which is, again, the best known, best studied. But right now, our water treatment, our water plants are not testing for other cyanobacterial toxins. So they're not testing for the neurotoxins, etc. Water is just one of the key ways that we get exposed to a wide variety of environmental pollutants and toxins. So anything that you can do to improve your quality of water, whether it's, charcoal filters or the options that you have, but these neurotoxins are not being tested for, other than microcystin.
Thank you. It looks like we have another question. You mentioned L-serine and its positive impact on the damage caused by the toxins. Is there a way that we can increase our production of the L-serine in some way as a protective factor? Hopefully I didn't butcher that too much.
Yeah, so that's a great question, and I'm glad you picked up on that because I didn't talk about it too much. In some areas of Japan, they have diets which are extremely rich in L-serine, and they have no cases of neurodegenerative disease like we see. And so some of our foods naturally have higher levels of L-serine, like eggs and bacon. But L-serine is it's a naturally occurring amino acid. You can actually buy it. Now, this is not something that I'm going to promote. I'm not a medical doctor, and I'm not here to promote that. Right now, they are in phased trials for L-serine as a treatment for those who are suffering from ALS. The initial trials showed such good results that they fast-tracked further trials of L-serine as a potential treatment or therapy. You can find those initial FDA clinical trials. You can find the initial results online and continue to follow up. I think right now they are in the midst of the phase 3 trial. So we should expect to see something coming out relatively quickly with that. But you can buy L-serine off of Amazon, and you can see in those FDA trials how they are using it as a food additive.
Thank you. Janet has a question. After listening to Dr. Zach Bush regarding COVID-19, he believes pollution is fueling the COVID virus, specifically the sinusides in the air. We live across from a CAFO and aquifer, sorry, can't read today, near an impaired river. Have any studies been done regarding the link between those neurotoxins and factory farms? It certainly seems like there's a huge link. Thoughts?
Well, so the link there would be the release of phosphorus and nitrogen from animal waste, or from, for example, if you had a large-scale industrial farm and there was the release of nitrogen and phosphorus into the water, and that nitrogen and phosphorus, either from animal waste or from fertilizers, if it gets into the water, then it fuels cyanobacterial blooms. So that is the link there. In regards to COVID, there is a link between general environmental degradation and large scale agriculture and the development of viruses in the mutation of viruses. But that's something I'd have to spend a little bit more time on. But yes, there is a link there between general environmental degradation and novel viruses.
Thank you. Scott has asked, if our own water treatment facilities are not checking for neurotoxins, how do we know if bottled water companies are checking for neurotoxins?
Well, at this point, I don't think any bottled water companies are testing. So this research is relatively new, and it certainly has not been incorporated into any large-scale infrastructure projects. So it's just going to take time for that to filter into how we treat our water and how we test our water and how we manage cyanobacterial blooms. So, the vast majority of the really good research that has come out has been in the last 20 years, maybe 25 years. And so, it takes a while for that to translate into changes in public policy or public health measures. So, yeah, at this point, we aren't seeing that testing, but hopefully we will soon as knowledge increases and filters down into our public utilities.
Great, thank you. Are there any additional questions before we end? Great. Kate, do you see any that I'm missing?
It looks like there might be one more. Scott asked if our own water treatment facilities are not. Oh, sorry, that was the one we just did. I just scrolled and saw it again.
Hi, I'm on the phone.
No,
Can you
I
hear
don't
me?
see any others.
Yes, we can.
Oh, okay. I have some questions. I actually have 3 of them. Okay. I guess the quickest one to answer would be, do you know if reverse osmosis water filters are effective for removing this?
Yeah, so that is something that we've talked about sort of amongst ourselves, right? What type of water treatment, and we see a lot desalination in the Middle East and things like that would potentially filter those out. And I don't have an answer for you yet. So that's something that we're talking about, that we're pursuing, others are pursuing, but I don't have an answer for you.
Okay. The next question is about that amino acid, serine or whatever it's called, L-serine.
Yep.
How would we know how much is worthwhile? I mean, it's like I might have 1 egg a week, maybe I need how many, do you know how much is needed of that?
Yes, yeah, I know exactly what you're asking, but I need to be careful in exactly what I say, right? It was a few, I know for the FDA trials, they're using grams, and you can simply Google those FDA trials. You will see the results, and you will see what people were taking. Again, this is now in phase 3. Everybody has their own unique medical situation, so I am not going to make any medical recommendations. I'm not a medical doctor. I'm a research scientist. But you yourself can look up those results, and you can discuss that with your healthcare practitioner, but also keep a very close eye on for the latest developments coming out in regards to L-serine. So it's L-serine, which we're seeing very good results from.
Okay, but if I go to the FDA trials, those are probably on people who are already ill. They're probably getting like massive doses, I would think, not something that I would take to help prevent problems.
Yeah. Yeah. So they did a series of doses in those trials. So if we can be, again, I want to be careful what I say. We will have more information coming out in the phase 3 trials, but again, you can find levels of foods which have much higher levels of L-serine.
Okay, so just like Google FDA trials on L-serine, is that what?
Yep.
Okay.
And much of that work was done out of Dartmouth, and the researcher that was head of that was Elijah Stommel, Stommel, Elijah Stommel out of Dartmouth. And so that's a good way. Otherwise, I can follow up, Laura. I can provide you guys with the links if you would like, and to some of these key papers if people are interested in pursuing that. I can provide you the links.
That would be great, and we can send it out.
So that sort of goes along with my last question. You mentioned we could get a copy of this. Would that be part of that, and how does that work?
Yep. So if you registered and gave an email when you registered, Kate,
Yep.
I believe you'll be sending that out. If you did not give an email and want to make sure we have your information, I posted a survey link from me in the chat box, and I will repost it again. You can fill that survey out about the program, and there's an option to ask for a certificate from the program, and we'll make sure all those people also get the recording.
Okay, so if you have my email, which you do, because I got information by email to get into this.
Perfect. Then you'll be getting it.
I'll be getting a copy, as well as what was mentioned about links that you just mentioned.
You'll be getting a copy of the recording, and then all of the links that Dr. Richter sends us, we'll make sure we forward on to you.
All right. Thank you very much.
We have a couple more questions that came in, and I'm not sure for Rob, is there a way to test yourself? Is that related to the water? Maybe we can get, assuming it's about the water, it came right after we were discussing water treatment.
So there are some labs that are testing. So yes, for example, let's say you wanted to test your own water for these types of neurotoxins. Yes, there are labs in the United States. I could send some links for those labs that are testing. But some people, as part of this early research, they had, for example, hair samples that were tested that actually showed accumulation of BMAA in hair and in protein. There's research going on now whether we could do a blood sample and pick up whether you have been exposed. So there's that possibility. And if you remind me, Laura, then I can provide the links for the labs that will test.
Thank you. We had another question on the same line, wondering if boiling water is better for us to drink. Does that help to remove the toxin?
So boiling water would be effective, for example, if you had E. coli, right, bacteria cells from human waste in water, or other types of bacterial contaminants. But it's not going to destroy the toxin. So in this case, boiling will not be effective. What I would like people to take away from this is not to be suddenly very nervous about their food, their water, but to generally have an increased awareness of how important our water quality is. I'll give you some examples. We're now starting to see the accumulation of these toxins in plants, our agricultural plants that have been irrigated with water, which have cyanobacterial blooms in it. So if we irrigate with water that has cyanobacteria, toxic cyanobacteria in it, we're seeing the accumulation in plants as well, in our agricultural products. So what we should be stepping back and saying, okay, water quality, it seems water quality is going to be fundamental to brain health. And I think we need to step forward and make sure that our public utilities, our public entities are ensuring that we have access to clean water. Because ultimately, without clean water, the food that we consume, whether it's animal or plant-based, will also potentially have toxins in it.
Thank you. We're also getting a lot of positive comments thanking you for all this information. Are there any additional comments? Kate, am I missing any?
I don't think so. That looks like all of them.
Once again, make sure that, and Kate, you're going to also send out the survey link when we send out all the information. So you can still get your certificate of attendance if you fill that out, or just give us feedback on the program. We are very thankful that you were able to attend with us. And thank you, Dr. Richard, for all your information.
Yeah, can I make one last comment, which I think is relevant now given COVID?
Yes, please.
In our research in the Middle East, what we found was that traditional dress, or the males and the females when they covered their face, reduced exposure as well. So something as simple as wearing a mask, it not only protects us from COVID, but in other situations as well will protect us from other types of environmental toxins which are airborne.
Thank you. That's a great point to add. Kate, do you have anything else before we end?
No, I do not. Just look for an email from me with the 3 things, the link, the recording, and then the links that Dr. Richer will share with us. And have a wonderful weekend, everyone.
Thank you so much. I'm so pleased to be here.
Thank you.
Thank you.
Bye-bye.
Bye.
Thank you.